All right, so the leaves of plants are incredibly diverse. We looked at just some basic ideas of how we examine the leaf and some of the terms that go with it. We looked at compound versus simple leaves, and the axillary bud is the key for that. And then we started looking at venation patterns. We have parallel venation, netted venation. We looked at stomata location, and we really thought more holistically about transpiration. Now that we're at the leaf tissue, we talked all the way through the plant body, how water moves through the plant. And then we started looking at leaf sections under the microscope, and that you can tell a fair bit about leaves under the microscope. So here I think we left off. And so this is a really nice image showing you many, many things. We looked at the idea that there's stomata on either side, top and bottom, so to speak, of this image. So that's reflecting, is that reflecting horizontal or vertical leaf orientation? Vertical. Okay, so it doesn't really matter where they place their stomata because they're going to get hit with about equal amounts of sun as the sun travels across the sky through sunrise to sunset. So it doesn't really matter. We looked at venation patterns. What is this venation pattern showing? Parallel or netted? No, netted. So this one, when the technician took the slice of the leaf, this vein was coming out at you. And this, believe it or not, is a vein. And that one got sliced, like my arm is the vein, it got sliced along the length when they took the cut. And so you have one vein going one way, one vein going the other, so that's netted. And then we cut this image in half and said top above the red line, below the red line. Pretty similar, right? And so we said this again is a characteristic of a leaf that spends its life more vertical than it does horizontal. Okay. What about this leaf? Parallel vein or netted? Parallel. So you have the major vein here, and then the two smaller veins, all kind of giving you the same look. So when they made that cut, they were all cut in cross section. Something else you can tell here. What are this ring of cells called? Yes, bundle sheath cells. So do those look skinny or plump? They're plump. So there's a particular way certain plants do their sugar production. And they really beef up their bundle sheath cells. Do you remember? Is it C3, C4, or CAM? What do you think? Yep, C4. So C4, we have a spatial segregation of where things happen. Certain things happen in the mesophyll cells, and then certain things happen in the bundle sheath. And because of this spatial segregation, they really do ramp up their bundle sheath cells, so they're much more plump, so they can fit everything in there because they have to run the Calvin cycle to really high levels in here because they're not really running it in most of the other cells. So this is a C4 plant. It's doing C4 type carbon fixation. And if you cut this one in half and look above the red line and below, it's pretty similar. So again, this is a leaf that spends most of its life vertical. So this is actually maize, corn. It's a corn leaf under the microscope. This is leaf. And they stained it so that this layer is really obvious up here. So what do you think that red stuff is? Yep, the cuticle. So that's wax buildup. Look at how thick that is. So if you were a plant and you devoted your resources to really thick wax over your leaves, what kind of environment do you think you might be living in? Desert, hot, okay, dry, and so this is definitely an example of that. And then let's cut it in half. Whoops. I want it cut it in half, kind of so red, it's hard to see my red line, but if you cut it in half and look top and bottom, they're pretty similar. So this is a plant that lives in a consistently dry area, probably drought prone, but its leaves are more vertical. It's hard to tell veination pattern because there's really no, you only see part of a vein here and that's it. So there's not a whole lot you can tell about that. This is a leaf, believe it or not. Not anything like what artists draw when they say draw a leaf. This is not what they choose to do, but this is leaf tissue. So look along the top here. Do you see any obvious stomata, guard cells with a pore? Mm-mm. How about right along this bottom? Do you see any obvious? Uh-uh. They're like, where the heck did they put their stomata? You got to go in. You got to go in. So let me blow it up. So you'll here. And here. So they tucked them up in the kind of inner part of their tissue. Is that to control water vapor? Yeah. So these are called, do I have let me go to here. That area that they're called stomatal crypts. Stomatal crypts, that invagination like that of the tissue where you see all the stomata are buried deep into the tissue area. So they're called stomatal crypts. And let me go back here. So now if you take this leaf and think about wind coursing along this leaf, and there's no stomata on the surface, there's no stomata here, they've tucked them up in here. So when wind goes across, are the stomata greatly affected by wind? No, they're sheltered. They're in a sheltered environment. So why would you want to shelter your stomata against wind or?
Slows down evaporation.
Yes, exactly. It slows down evaporation. So this is actually, if you measure the relative humidity in these stomatal crypts, it's quite high. Remember, anytime you have really high humidity around a leaf, that slows down transpiration rates. So this is a plant that must be living in an area that it has to worry, so to speak, about too fast of transpiration rates. And so one way to slow it down is to tuck them in like this into a relatively high humidity stomatal crypt area, sheltered from direct wind. And so maybe it is a windy environment. You know, it could be a consistently windy environment too, that they choose this design through adaptation. So this idea of, you know, probably living in an area that's drought prone, maybe consistently windy. And so they're going for a pretty dramatic design here. So the fancy word is for this type of plant, where they're living in general, the habitat that they're living is xerophytic, all one word, xerophytic. Zero referring to dry. Anytime you see PHYT in a science word, that means something relating to plants. So it's a plant that likes or is adapted to dry environments, xerophytic. So pretty cool design to tuck the stomata like that. Now, if we go back to the previous, is this a monocot or dicot, do you think? Can you suss out the vascular tissue? Monocot. Yeah, monocot. Monocot, all cut and cross section. It's oleander. Oleander. So yeah, really funky design, but effective. Some even say, oh yeah, I'll put some trichomes in the stomatal crypts. I'm gonna one up you. So this even slows down transpiration even more because that creates, it's harder for the air molecules to get stirred up by any wind if you have those trichomes in the way. So this really creates really low water or air molecule movement in here. And so again, really slowing down transpiration. And look, they even have quite a nice layer of wax, Lacutin wax, top and bottom. So this is a nerium leaf, Looks like this leaf spends most of its life horizontal. The way if you cut it in half, they kind of look different. This leaf, again, is pretty weird. So this is a gymnosperm needle, like a pine needle. And technically they're not leaf tissue, they're actually modified shoots, but it's understandable why people call them leaves. So they don't have stomatal crypts, but they have what we call recessed stomata, recessed. And so, leaves, pine leaves, stay, they're evergreen through the winter. Evergreen trees, conifer trees can go dormant for stretches of time when it's too cold, but when, like today, the conifer trees are active. They are waking up and doing a little bit of physiology, so they will need to have their stomata open for bringing in some CO2. But they kind of tuck them down just a little bit because in the winter, even though there's plenty of snow, it's still dry for them. It's a relatively dry time period in their life cycle because the water's all frozen. It's frozen in the ground as well as the snow on top. So they don't have a lot of access to water. The vascular tissue, you just see two bundles right here of vascular tissue. So needles are pretty thin. They don't have a lot of space for that. Now, deciduous trees, plants, in the fall, they get rid of their leaves. So this is the abscission zone that they have to form in order to get that to happen. And so it's in the petiole region of the leaf. So remember the leaf, you have the leaf blade, and then the petiole that attaches the leaf blade to the stem. So it's in the petiole region that you'll have the abscission zone forming. So it's a layer of cells in a line that the cell walls start to weaken. And after a while, once that whole line of cells weakens enough, then the petiole falls off, the leaf falls off, and then they kind of suberize that area so that you can't get easy entry by pathogens. So the suberin is wax that they'll use to make sure that it's a little bit beefed up for protection and to prevent getting dehydrated. So under the microscope, you would see something like this. So you'll look at that this week in lab, where you can see the abscission zone all along there forming. And so it's pretty effective method. Here's another image. Right there is the abscission zone. So during the summer, when things are cooking along just fine, the hormones in the leaf are pretty much signaling to the plant, we're healthy, we're vigorous. So auxins and cytokinins will be giving that signal. There'll be enough of those hormones there, and you can see that they flow down. And so they're telling the suberization, or the incision area, everything's good, we're healthy, we're vigorous, we're vital, we're doing our thing. And then as fall starts to roll around, due to a lot of signals the plant is interpreting, their oxygen levels start to drop, and that will tell them physiologically, okay, it's time to ramp down. Let's collect all of these useful molecules down into our roots, get everything out that we can, and then start shedding the leaves through leaf abscission. And so you tend to see cytokinins and auxin levels go down and ethylene come up. And it's really the ethylene that's doing the final, okay, form the abscission zone and fall off. So there's a lot going on here, but ultimately it's that abscission zone that will allow for the leaves to fall off in the fall. This is a leaf. Pretty funky. You gotta really look closely here for the vascular tissue. Not much. Hardly any xylem, just a handful. Small cluster of xylem. What do you think this area? I mean, it's not any cells in there. What's in there? Air, just air. So why would you be a leaf and want lots of air inside you? Yeah, to float. I want to float. So it's like having your little life preserver built into your tissue. And another dead giveaway is there's hardly any xylem. But if you're just swimming around in water, I guess you don't need a lot of xylem. So don't devote so much to xylem. Make sure you have plenty of air pockets so you can float. So this plant's probably floating on the surface, not one that's submerged all the way. It's probably designed, and it is, it's a water lily. It's designed to float. And so you can see it spends its life horizontal, floating on the water, and you can see it's a very different image, not a symmetrical look to it above and below the halfway point. So yes, this is a water lily that likes to float. Yes. Yeah, it looks like maybe one right here. Maybe there. It's hard to see. Maybe there. Hard to see. Not all, when the technicians take the samples, sometimes it's hard to capture everything. Yeah, that's like when you looked at slides, was it the first week when you looked for, oh my goodness, it was a crystal, and it was like it dangles down in yes, cytoliths, thank you. That is so hard to buy slides that have that visible, because it's just really hard to capture that when they take sections. So we have a handful of slides that can show that, but it's really challenging. Anyways, so yeah, water lily, they have wonderful architecture underneath too that you don't often see unless you really go kayaking near one and look. We don't have them this big where we're located here, but we still have some interesting lilies, especially on inland lakes here. Okay, so leaves can look very different from what you typically see, especially from an artistic rendering of a leaf. So, and again, it's driven by what kind of environment are they adapted to. And so you can see that they have changed their leaf morphology depending on where they live. And so it's, you know, that selective pressure of their environment really driving how they're going to design their leaves. And then even like we said, you know, there's shade leaves and sun leaves. So some plants will have different leaf types on the same plant, you know, so they can vary it in just in their lifetime, what the leaves will look like if they're a big enough plant where part of them are in the sun and part of their body is in the shade. So really remarkable part of the plant, lots of diversity in the morphology. Okay, so this is where you can draw the line for the exam material for this Friday. We won't go any further than this for this Friday's exam. So it will cover photosynthesis and all of that. We covered on that, and including, you know, the PCO cycle, Kelvin, C4 CAM, and then root stems and part of the leaf content through this slide set. But I would like to get into the next slide set on leaves as well. So that's part 2. And we're going to look at some modifications, interesting modifications. So you don't have to write all this down. This is part of the PowerPoint, but I'm going to show you these ideas in terms of how they can adapt in some really interesting adaptations, depending again on where they're native to, what they've adapted to. And so we already talked about tendrils in the stem tissue PowerPoint. So there are tendrils that are based from stem tissue, and then there are tendrils that are leaf tissue. So it's a little confusing. So, you know, plants are amazing that way. They can do what they have to do depending on their environment. Spines, we kind of looked at thorns for stems. We said that some thorns arise from stem tissue. Now we have spines, which are very similar, but that's technically leaf tissue. All right, so it's a little confusing. And then I'll go and work through some of these. We've already talked a little bit about carnivorous plant adaptations and cotyledons as part of the embryonic plant inside the seed. So we'll look at these. So we already talked about this too, that leaf tissue can be used, not just this one. There's many, many leaves that can be used to take cuttings and propagate plants. The Sansevieria or snake plant is a good one for that, but you have to be patient. So if you did take a cutting of this, you have to be patient. It's going to take a while, but then they're really hard to kill. I mean, they are just, I had 3 in my house that has very poor lighting and they didn't care. They're like, yeah, whatever, you know, so they're really, and I forget to water them and they're just fine. So here you have a pea plant that's like, help me, I need to get upright. And so they will modify some of their leaf tissue so that they can reach for a solid surface around them and hopefully be able to get their stem and leaves upright enough for photosynthesis. Here's another example. Here are some spines. So it's a little confusing because thorns versus spines, but again, you know, same basic idea. In this particular case, this plant says, well, I'm going to modify my leaf tissue to be pokey so I can hopefully discourage herbivores coming along and trying to eat.
Picture practice that was for the plant.
Mm-hmm.
That main like paddle shape is the stems.
Yeah. Yes, you got it. So the cladophil is a flattened stem, and then the spines are the modified leaf. So yeah, when you look at a lot of desert plants, they don't have nice thin fleshy leaves, you know, the real thin ones. And if they do, they sure have some way to defend themselves, usually pokey things. But they can also have some pretty potent toxins. So yeah, I mean, when you look at the plant body, the most vulnerable part of the plant body in terms of getting eaten is the leaves, because they're thin, they're full of nutrients, all that sugar getting produced. So here, again, another succulent. This isn't even a cladophyll, it's just kind of nice plump stem tissue. But the leaves have just been devoted to, I'm just gonna make spines instead of leaves so that I can protect, because a lot of what's going on in the stem tissue is not only sugar flowing through there, but also water. And so now the stem is the one that's devoted to photosynthesis. The spines aren't. It's the stem tissue. So it's not always just leaf tissue that you have photosynthesis. So we have buds that are modified leaves for either the axillary bud, or you have terminal bud scale scars, they call them, where the leaves fell off. You have overwintering buds that are waiting until spring before they'll leaf out. And so protection mode, definitely, with the buds right now on the trees. So those are definitely modified leaves to kind of shelter and protect the leaves underneath. These are leaves. I saw this in a store locally. I'm trying to remember where I saw this. This is called the window leaf plant. So if you were to see this in its natural setting, it'd be a desert, and it would be all buried except for just the very surface. That would be poking through the soil. And so in a normal setting, it's mostly buried. And then the kind of this conical shape, it kind of narrows down and kind of, as you go up, it kind of flares out like that. So it has a nice surface area. So that's the part that will be poking through the soil. All the rest is buried. So why would you bury yourself? Where do you think this plant might live? Really hot. Yeah. So this is a succulent, nice fleshy leaves. The internal working of the leaves here, the way the internal cells are designed, it's super, they've done like the physics of it. So when you shine light or the sun is beating down on this plant, the way the light bounces around inside that really succulent leaf is just amazing. They really have a design that optimizes scattering of light and bouncing around so that all the cells there, all the way to the depth of that leaf, will get their fair share of sunlight for photosynthesis. And then, yeah, they bury most of their body because it's bloody hot in the desert. And so this is one way to stay cooler. It's also a protection mechanism so they don't get chewed on, because you can see there's no spines or thorns or anything. So it's like, okay, we have to figure out a way to stay sheltered from predation. So they just they're buried. Beautiful with the flowers there blooming. We've talked about this before, how the poinsettia flowers, the red, are really modified leaves. They're called bracts. Flowers are these teeny tiny things, but taken as a whole, you can understand why people say, oh, look at the poinsettia flowers. And they include the red bracts. Definitely effective. And if you think about it, the leaf tissue sticks around a lot longer than flower petals. So this is a plant that's like long-term advertising. It can have those bracts persist much longer than the typical flower petal persists. So it's a good strategy. You find a lot of tropical plants will do that. They'll turn some of their leaf tissue as an advertisement, and they just have a longer stretch of advertising because their petals, if they did flower petals, those tend to not last as long. Very colorful. What's this one? Aloe. So again, good storage. You worked with aloe at least once or twice in lab, and in the lab manual, I think it does talk a little bit about the chemical that you can rub on wounds, for example. I always try to keep an aloe in my kitchen. I burn my hands a lot in the oven, touching the oven or the burner accidentally, so it does work. I had a student take this class several years ago, and he was a chef in a local restaurant, and he got really nasty burn on his forearm, and he just like 3 times a day he would break open the aloe plant and just smear that, and he kept taking photos of his arm injury, and I couldn't believe how quickly it healed and no scar. It was dramatic. So that was proof of concept for sure. This is interesting. So this is the left image is how it normally is. The right image, they took it apart so we could see what's inside. So these are leaves, modified leaves. They're called flower pot leaves because they kind of seal along the edges and create a pot-like configuration. And then they put roots down into it. And the reason they do that is that you can kind of see there's some debris in here. And so there's like, twigs or little bits of stuff that will fall in there. And then insects, especially ants, like to go in there and kind of hide out. And if they die, their body's full of nitrogen and so on. And so they capitalize on stuff that's collecting in there. So this is a tropical plant that has adapted to trying to catch things in its flower pot leaves. Anyone play around with this one? So mother of thousands, a kalanchoe species. So cute. And so you have all along the leaf is points of cloning. These are what we call plantlets, and you can even see the roots forming. So, you can imagine if an animal brushed by that or water, raindrops, you could see how those would quite easily break off, hopefully fall to a suitable soil area and start growing. Here's a really nice image. So quite an interesting adaptation, very good. I mean, we used to have a lot more mother of thousands plants in the greenhouse, and then that was back in the day. Some of the students, one of my graduate students decided to go in there and water with the hose, and he just went, and knocked all these off, and pretty soon, almost in every pot, we had mother of thousands plants everywhere. It's like, no, don't water like that. We had to tell them, stop doing that. It was a very hot day, and the temperatures in the greenhouse was way too hot. So it was a good strategy. He tried to create a nice cooling effect, but it caused the mother of thousands to really disperse. So here, this is a oak seedling that's germinated in a natural setting, and you can see how plump the cotyledons are. So all that tissue is cotyledon. So really full of plenty of nutrients to keep that plant going until the leaves, the true leaves, are up and running so they can do their own photosynthesis. Can you tell what these are? Yeah, pitcher plants. I don't think we have these species here, but they're really cool. Do we?
Yeah.
On the left?
It's like little purple.
And I know the purple ones. So cool. So they have hairs inside there that point down. So when the insects get in there, they can't really climb up. The hairs, the trichomes get in the way. And then there's liquid that collects. And so when the insects get in there and can't make it back out, they usually fall and drown. And then there's all kinds of microbes in the liquid that help the plant digest the insects so that they can get access to the nutrients. Aren't these beautiful? Sundew And we don't have that. I can't, that must be a find if you stumble on that when you're out hiking. My goodness. Beautiful. And of course, modified leaves, highly modified leaves to trap insects, the Venus flytrap. This is how they say plants can count because the insect has to walk and trigger 2 trichomes in a modified leaf before the leaves will snap shut. Very effective. There's a Sir David Attenborough video where they put music to when the shuts and it makes it sound like the fly squealing in pain. It's like, oh, you feel sorry for the fly the way they put the music to it. Here, I think you can see all the yellow, those are called Beltian bodies. It's very rich in lipids, and it's a trick that a lot of acacia species do to lure ants to, hey, come hang out with me. And the ants will form nests in some of the thorns, and then they'll patrol the acacia plant, keeping other insects off of it that are trying to chew the plant, and then they feed on the Beltian bodies as a reward. Video of this is pretty intense when if they probably, dropped some hapless insects on the acacia, and then you wait to see all the ants just come and swarm, and they'll fight with that insect and actually like knock it off the leaves, or try to bite at it and get it to fly away. It's impressive. So that acacias pretty much have recruited an army to help protect against other insects that want to try to come along and chew their leaves. Because, their leaves are fairly vulnerable in their design. So they recruited their own army. So pretty effective. Okay, I'm going to stop there. We still have some more things to say about leaf adaptations and how they defend themselves, but we'll save that for Wednesday. So, okay, we have let's go back to the photosynthesis material. I think I'm going to show different screen here. And do this. All right. So what questions, I think we should focus on this today. What questions do we have on photosynthesis? We have both the light reactions, and then we spent more time on the Calvin cycle and other adaptations to that. What kinds of questions do you have? Yes.
It's got the light reactions, and we got like the biochemical reaction / carbon fixation. It's like the Calvin cycle, the C4 cycle, the CAM cycle. Are those all under like the carbon fixation?
Yes. It's just the C4 and CAM cycles. I like to think of them as like additions to the C3 cycle, like they get tacked on to the C3 cycle to help them concentrate the CO2. So they're trying to concentrate CO2 around a particular enzyme. What enzyme? What enzyme is sloppy and can't really tell the difference? Rubisco. All right. So the CAM and C4 are designed to really make sure that Rubisco sees carbon dioxide. Very, very little or no oxygen. It looked, when I was grading, hand grading the assignments, it looked like most of you were clear on what is RUBP versus Rubisco. So that was good. Yes, Taylor.
What's the PSI 1 and PS1 and PS2?
Yeah. So Yeah, let's see. Oh, here. Photosystem 1 and photosystem 2, PS1, PS2. Basically, when we consider the light reactions, just know that they're named photosystems because they will be absorbing light energy. And it's in those photosystems that you'll get a chlorophyll's electron excited by that light energy. All right, so that's what they're showing here. They're trying to show that there's a very special chlorophyll here in photosystem 2, that when photosystem 2 absorbs that light energy, that electron gets excited and then gets sent along this electron transport chain. And then the same thing's happening in photosystem 1. There's a very special chlorophyll here that when light energy reaches that chlorophyll, the electron gets excited. And so having both those photosystem in play, make sure that you have this nice electron flow. And the goal here is kind of twofold. They're trying to get this electron transport chain process going so that they can get the terminal electron acceptor reduced. So NADP plus will get that electron and get reduced to NADPH. So that's great. That's one of the goals. And then the other goal is to figure out how to tie this electron flow to making some ATP. And so they're doing that by the idea of getting a proton gradient established. So key parts of this process allows for protons to build up in the lumen of the thylakoid. And anytime you get a proton gradient across the membrane, that's a really good situation to make some ATP. And so that's what they're doing here. So it's twofold. The light reactions really have, two major things happening. You get reduction of NADP plus to NADPH. You get a nice proton gradient, which will give you that ATP. This is, you're doing a similar thing in your mitochondria right now. It's just, unfortunately, we can't use light energy. We can't do that. We have to eat something. That's our energy source. Other questions on the light reactions? What about Calvin cycle, PCO cycle, Rubisco? Yes, Taylor.
So I did, I was wondering, like, when we talk about the 19 families of plants, and then we talk about different different genus.
Yep.
That, are we gonna have to know that for this?
No, not to that level. So I was trying to show that basically if you understand, where did it go? Like here, with the CAM, not all species in one family do it, lots of independent evolutions. So just trying to get across the point that this CAM design has popped up many times in the evolutionary process. It's not just, one plant that figured it out and everything, went from there. It's pretty diverse, you see a different plant family will have all kinds. Some just do C3, some do CAM, some do C4. So that's another indication that this has been popping up independently over time. And the C4 cycles like that too. So one thing that several of you missed on your assignment was what is the sugar made? So that will be on the exam. So that's this slide. So the sugar that's being made that the plants care about is right there, sucrose. So this is the Calvin cycle is happening in the chloroplast. And then if they need to make sugar, they'll send out some of the G3P into the cytoplasm to make sucrose. If they have plenty of sucrose made and cooking along, then they might say, oh, let's just make starch instead. And so they have this either or option in that cell. They're like, should I make sucrose and send it along, or should I go ahead and save up and make starch? Well, so you have sucrose is a disaccharide. It's fructose and glucose put together.
Oh, so it's the both of them together.
Yeah,
Okay.
so sucrose. So the reason plants prefer sucrose, they want a disaccharide. Because when it's transporting through their body, it's less likely to react with other things along the way. So it'll reach its destination if it's a disaccharide. And yet when you cleave that, it's a good amount of energy that gets cleaved when you break the fructose glucose bond. So sucrose, sucrose, that's gonna show up on the exam. Guarantee it. All right, we'll reserve time Wednesday, a little bit more time on Wednesday compared to today for last minute clarifications.